This you will understand with the help of this picture better. So the normal structure of hemoglobin, when oxygen comes and binds to the hemoglobin, so the tightly bound structure of the hemoglobin starts opening up when one molecule binds. Then second molecules also gets ready. Third also comes. Fourth also comes. So the taut form, taut means tight form of the hemoglobin which was closed, it is now opening and it is converting into relaxed form and it is binding with four molecules of oxygen. That means one molecule of hemoglobin has an affinity for four molecules of oxygen. It can carry four molecules of oxygen. That is the meaning beta. This is oxygen dissociation curve. Now see here, you see this is the partial pressure of oxygen in the tissues. Only initial binding of the taut form to oxygen will take more time. Okay, so once it is binding with oxygen molecule, remaining three oxygen molecules will come and bind immediately. So this is the dissociation curve. This is given. So percentage saturation with oxygen. So this is percentage of oxygen concentration in the atmosphere. So this is hemoglobin saturation. So only hemoglobin saturation, only one molecule of oxygen, it is difficult. Later all the three molecules will come and bind to the hemoglobin molecule very fast. This process is taking place in the lungs. And in myoglobin means what? The hemoglobin which is present in the muscle is called as myoglobin. The percentage saturation of myoglobin is still at a faster rate when you compare with the hemoglobin present in RBC because skeletal muscles are actively contracting. Read it once better, you will definitely understand. Mucosal block theory of iron metabolism, we will check in the internet. I'm sharing here in our group beta. Check once. So mucosal block describes ability of initial dose of ingested iron to block absorption of second dose given 2 to 4 hours later. So mucosal block, that means once we are eating iron in the diet, so after 2 to 4 hours only second dose of iron can get absorbed. Till then it gets blocked how much ever iron we take in the diet. So the studies indicate that mucosal block is fast responsive endocytic mechanism destined to decrease intestinal iron absorption during high ingestion of iron. Okay, it's very much clear beta. So if you are taking iron in the diet for 2 to 4 hours, even if you take excess iron, it won't be absorbed till that initial iron is completely absorbed. That is the mucosal block theory. Okay. These are simple things only beta. Boyer binding mechanism. Can you share this, Niharika, till I go to the next answer? Because
Yes, ma'am.
yeah, please. Protein electrophoresis is what we have discussed with paper electrophoresis. That is same beta. G proteins we will discuss. Till you find that answer, I am checking with the next answer. Okay. See here, write about the bonds which stabilize the structure of protein. So bonds which stabilize the structure of protein, you can see here we have covalent bonds, we have sulfur bonds, we have van der Waals forces, we have hydrogen bonds. Many bonds are there which will keep the structure very stable. There is a question like biologically important peptides here also, which we already did. I'm showing you here because we did it. Peptides which are less than 10 amino acids. Glutathione, many are there. Oxytocin. See here, beta. Here you have the answer. What are the various bonds? See, which are stabilizing structure of protein. So we, in the protein structure, we have two types of bonds. One is covalent bond and non-covalent bond. Covalent bond means the peptide and disulfide bonds which we see in protein structure. Okay, like they are strong covalent bonds, and their characteristics we will see. Disulfide bonds also I'll show you. Under non-covalent bonds, we have hydrogen bonds. Okay, we have hydrophobic bonds. The non-polar side chains of neutral amino acids tend to be like acidic, no, no, closely associated with each other. We will see this. All these bonds we will see and learn. Electrostatic bonds are there, van der Waals forces are there. All these things you are supposed to write. Let us start with covalent bond. See, covalent bond formation, how it is occurring, we will see the formation of peptide bond. See, this is the formation of peptide bond, which is a covalent bond. You are seeing amino acid 1, amino acid 2. This is carboxy group and this is amino group. So if water molecule is removed, one peptide bond is formed between these two. You call it as one covalent bond. So apart from covalent bond, when you see that helical structure, this one, so you are finding various other bonds, like you have hydrogen bonds between the chains. Okay, this is one hydrogen bonds. We also have disulfide bonds, you can see between cysteine and isoleucine. Like you have seen, like two adjacent chains, polypeptide chains, sulfide bonds are bonded. This is disulfide bonds. Okay, if you see this is also one alpha chain, one beta chain. Sulfide bonds are found. Okay, those are disulfide bonds. Okay, so with the help of pictures you will understand better. So those are the various bonds: hydrogen, disulfide, peptide, and the weak forces what we see between these like peptide bonds and protein chain. Those are van der Waals forces. Okay. If you are ready with answer, you can share with it.
Ma'am, actually, even I don't know that answer, ma'am. That day also in exam, I did not write that answer.
Okay, okay. Kirti, you have written that answer, no? Can you share that? Boyer binding mechanism. Even Rohita, you also have written that, I remember. I'll take you to G proteins meanwhile.
Okay, ma'am. I'll share.
Thank you so much.
Okay. Okay.
Hormones chapter we will go with.
మ్యామ్ నేను ఎగ్జామ్లో క్వశ్చన్ స్కిప్ చేశాను మ్యామ్. వైర్ బైండింగ్ మెకానిజం. ఓకే.
No problem, beta, don't worry.
ఓకే మ్యామ్.
Okay. Now see here, first we are asked definition about G proteins, beta. So I am giving you the information in our group. So then we will talk how it is related to hormones. So please read there, beta. I have shared just now. See, I am reading. G proteins are also known as guanine nucleotide binding proteins, like guanine nucleotide binding protein. Okay, and these are family of proteins that act as molecular switches inside cells. That means if you want to transfer something, see, this is cytosol and this is outside. If you want to bring hormone from outside to inside, generally our body makes use of these G proteins. That is the meaning. Molecular switches inside cells and are involved in transmitting signals from variety of stimuli outside the cells. Like G proteins, example we have enzymes GTPases. Okay, let us see now. Now see, I'll include two answers here, beta. Very simple only, it will not take much time. Mechanism of action of hormone, we have two types, beta. Mechanism of action, this is steroid hormone. Okay, so this is group 1 hormones, they are called as. Like we have two types, group 1 and group 2. See here, we have group 1 hormones and group 2 hormones. Group 1 hormones, they act in one way, and group 2 hormones, they generally act with this GTP, G receptor, especially second messengers they make use of. Okay, so like CAMP, GTP and all, they come under group 2. Now let us see group 1 first. How are they acting? So group 1 hormones, like they will have, suppose this hormone is growth hormone. Okay, so you will see it is entering into the like cell. So this will have receptors inside the cytoplasm, and once it is entering into the cytoplasm, it is entering, the hormone is allowed to go into the nucleus, and we know it in the nucleus it will be undergoing transcription, translation, and it helps in the formation of specified protein. The same thing we studied for vitamin A, vitamin D, same type of thing. That's why vitamin A and vitamin D, they are nowadays they are called as more of hormones than they are called as vitamins. Okay, so this is the mechanism of action of group 1 hormones. Now for group 2, they are actually acting through second messenger, cyclic AMP. So under this we have G proteins also. The process is actually mediated by G proteins, so designated due to their ability to bind with guanine nucleotides. In the definition of G protein, I told you. So second, group 2, they act through cyclic AMP. Let us see how do they act. Okay, this is group 2, mechanism of action of cyclic AMP. This is the mechanism of action of group 2 beta. Now let us see how it is acting. See, this is cell membrane. So on the cell membrane, we have certain receptors. Okay, so like the hormone comes and binds to this with the help of this G protein receptor, GTP, guanine triphosphate receptor protein. So it is now, this hormone is now allowed to enter inside the cytoplasm. And once the hormone enters into the cytoplasm, again it will do the action of like transcription and translation, but in a different way. Okay, here ATP is converted to cyclic AMP. Okay, ATP is losing its high energy phosphate, and this cyclic AMP, like AMP, is now converted to AMP with the help of phosphodiesterase. Like through series of steps, you will see whatever we want, the biochemical response is being brought about by this G coupled protein. Okay, that is G protein. Rohita, did you find that answer, beta? Just I need that page. Like which type of enzymes is your last question, no? Question number 20. There are certain enzymes which we can use to treat certain diseases. Like enzyme streptokinase can be used to remove blood clots. Aspergillus we can use in cancer therapy. The pain enzyme we can use to stop the inflammation. Okay, like that. Many enzymes are there. You can use the enzymes. Yes, please share it. Expectation of physician is a general question. Breakdown pathways are catabolic pathways and synthetic pathways are anabolic pathways. So under anabolic, you can talk synthesis of anything like glycogen synthesis, one example.
Ma'am, I shared.
Okay, nice, very good, beta. See, what are peroxisomes? See, generally peroxisomes we find inside the cells. What you have been drawing since your childhood, peroxisomes. Generally, we see inside the cytoplasm. So they are spherical or oval in shape. So inside the peroxisomes, we contain enzymes like catalases. Okay, so catalases they protect the cells from toxic effects of hydrogen peroxide. Okay, that means like oxidation is prevented. But just read once. I'll just take a small break. College people are going. So that is better. They help in actually what we have studied as antioxidants. No, like they will be neutralizing H2O2, O2 minus all. So similarly, even this peroxisomes also work in the same way. They just neutralize H2O2. Okay, so peroxisomes are also involved in the oxidation of long chain fatty acids and synthesis of plasmalogens and glycolipids. Plant contains glyoxysomes, which are special peroxisomes involved in glyoxylide pathway. So this much is enough better. Okay, peroxisomes. Anyhow, it is for 307. No? Thank you so much.
Ma'am, can I share, ma'am?
Yeah, please. Better. Niharika, meanwhile, acute phase reactant proteins, can you please share? Or
Yes,
I will
ma'am.
share it with you. No problem. Ah,
Hello, ma'am. Ma'am, it is of 3 pages, ma'am. Matter is in 1 page and diagram is another page, ma'am.
okay. It's better I share. Okay, Paul Boyer. Okay, rotary motor model of it. Okay, this way you can't understand better. So I need to explain you. Thank you so much for sharing. I will open and I will explain you. Okay,
Okay, ma'am.
I'll open the page and explain you better. Thank
Okay,
you
ma'am.
so much. We have done chemiosmotic hypothesis, right? Similarly, this is what you are sharing, right? Rohita? So, rotary motor model for ATP generation. For ATP generation, what is the model we have like understood till now? Chemiosmotic hypothesis. But there is one more model which is called as rotary model. Beta, let us see what is that. So it was proposed by this Paul Boyer. That is conformational change in the mitochondrial membrane proteins leads to the synthesis of ATP. Okay, so this is the figure. So in the mitochondrial membrane, you will see inner mitochondrial membrane. This is outside, this is inside. So some conformational changes will occur because of the entry of hydrogen ions. Because of the entry of these hydrogen ions, you will see same thing, coupling of ADP with high energy phosphate to form ATP. Okay, so in this, let us see what he has told. See mitochondrial membrane, so this is leading to the formation of ATP. So the original Boyer hypothesis now considered as rotary motor engine driving model. Okay, so the enzyme ATP synthase, here we have this F0, F1. So complex of enzyme complex 5, F0 subcomplex is composed of channel protein C. This is better. F0 is, it is made up of these C subunits, to which F1, this one is F1. F1 ATP synthase is attached. You just see this picture, you will easily understand. ATP synthase is coming and attaching here, F1. So F1 ATP synthase consists of these subunits. See, alpha, beta, gamma. Okay, this gamma subunit first, he says, surrounded by alternating alpha, beta subunits. So in response to proton flux, that means hydrogen ions are moving inside. No, because of the entry of hydrogen ions, you are finding there is a coupling of that ADP with high energy phosphate to form ATP. So this is what, according to binding changes mechanism, three subunits. F1 ATP synthase adopts different conformation. Now again, this is also given beta. See, this LOT is written. One subunit's name is loose form, tight form, and open form. Okay, one is gamma, gamma, gamma. So with the help of this, you will see ADP is coupled, ADP is coupled, ADP is coupled to form ATP. Here one ATP, one ATP, one ATP. So the model is different. So this is something like rotary model. So draw this model first. Because of the presence of ATP synthase, it is getting opened up like this and it is allowing ADP to couple with ATP. Okay. So this is the model which is being discussed here. Okay. So Niharika, are you ready with the answer?
Ma'am, acute phase proteins and the question are the same, ma'am.
This is not there in textbook. No problem. Is it there or not, beta? Acute phase reactant proteins. Actually,
Reactant, what is not there, ma'am? Acute phase proteins are proteins I found, but acute phase reactant proteins I did not find, ma'am.
okay, okay. See, basically, acute phase reactant proteins means what? We generally, we will have some inflammation, right? Like, suppose if we have some injury, I told you when I was doing glucocorticoids, when we have some injury, we will have the release of so acute phase proteins. Okay, you're sharing. Refers to non-specific response to stimulus. That's what beta. We have studied inflammation, no, when we were doing glucocorticoids. Just a minute, beta. Again, somebody is calling from my college. See here, beta. So, acute phase proteins refers to non-specific response to stimulus of infection, injury, various inflammatory conditions. Here you have to write the list of proteins which are released. We have alpha antitrypsin, alpha 2 macroglobulin, ceruloplasmin. Okay. Not only this, we also have this C-reactive protein, beta, and we also have many other things like histamine, serotonin, bradykinin, tumor necrosis factor. Many things are there which are released in response to inflammation. So you can write this, but not a problem. Okay. Yeah. You can write till here, till coronary artery disease. This is enough for this question. Okay, nice. Are we left out with anything, beta? Just check. Oxygen walls, visual, block theory, boy. Inhibitors of ATP synthesis, we did know better when we were doing electron transport chain. You can write the same thing. British anti-Lewisite, isn't it? Abnormal proteins. G protein. Okay, beta, we are done. So what is the question here? Like generally society, that is easy. Catabolic means what? Synthesis. So you can write glycolysis as the breakdown mechanism of glucose, that is catabolic. Anabolic, some protein synthesis you can write better, that is anabolic pathway. That means all amino acids are joining together to form some protein, like that you can do. Okay, so we will stop here. So tomorrow actually I need to go to college better, so I will be able to take only after 7 p.m. So please cooperate whenever I am not here. Okay. So see you all tomorrow. Bye beta, study well. This week you will be writing physiology paper, no? So whatever we have already discussed, you just share that physiology paper. Today's paper is worst paper, I don't want. So Niharika, you select one paper and share it so that everybody studies that, and solved paper you share and we will write it on Sunday. Okay?
Paper, ma'am.
Whatever you feel like is standard, till now whatever మనం సాల్వ్ చేసిన పేపర్స్ లో ఏదో ఒక పేపర్ ను మంచిది సెలెక్ట్ ఆల్రెడీ మనం ఫిజియాలజీ పేపర్ సాల్వ్ చేసాం కదా బేటా, దాంట్లో మంచి పేపర్ నువ్వు షేర్ చేస్తే సండే రోజు మేము రాసేది.
Okay, ma'am. Fine, ma'am.
Okay.
Yes, ma'am.
Okay, beta. See you all. Bye.